Hello friends, today we will study about a very important topic, namely electron transport chain and oxidative phosphorylation. You will get at least one question from this topic in your exam. It's very important. See, in glycolysis, glucose is converted to pyruvic acid. There, in addition to ATP, we will get the reduced coenzyme NADH. Similarly, pyruvic acid in Krebs cycle, pyruvic acid for the first time it is converted into acetyl coenzyme A by oxidative decarboxylation process. There also we get the reduced coenzyme NADH, and that further oxidized in the Krebs cycle, there also will get NADH and FADH2 in addition to the ATP, that is GTP, GTP is equal to ATP. In electron transport chain, we focus mainly on these reduced coenzymes because electron transport chain is all about this reduced coenzyme. This NADH and FADH2 are electron carriers. During electron transport chain, these reduced coenzymes NADH and FADH2 get oxidized and energy will be released. And during oxidative phosphorylation, the ATP is formed from ADP by utilizing the energy released from this reduced coenzymes by their oxidation. Now we can study about the electron transport chain. You know, this is the mitochondria. Here the glycolysis take place in the cytoplasm, glycolysis. Then Krebs cycle in the matrix of TCA cycle that take place in the matrix, and this is the outer membrane, this is the inner membrane. Here it is in the mitochondrial membrane, that is inner membrane, that the electron transport chain take place. It is the inner membrane space. Okay. Electron transport chain consists of 5 electron carrier complexes. This is complex 1, complex 2, 3, 4, 5. So, when electrons are carried from one electron carrier to the other, it is coupled with the formation of ATP with the help of the fifth complex, that is ATP synthase. This ATP synthase enzyme, it is protruding towards the matrix from the inner membrane. In electron transport chain, or it involves a series of oxidation reduction reactions in which there is a suitable electron donor and a suitable electron acceptor. Thus, there is a redox potential. The redox potential is a measure of the affinity of a substance for electrons. In electron transport chain, actually what takes place, the reduced coenzyme get oxidized. Here, first of all, the NADH, the reduced coenzyme get oxidized with the help of enzyme NADH dehydrogenase enzyme complex 1. Then the electrons are transferred to FMN, the prosthetic group of NADH dehydrogenase enzyme. So, FMN is part of NADH dehydrogenase enzyme. From the FMN to the iron sulfur protein, then through their ubiquinone. Then proton from this NADH is passed into the intermembrane space. This is the intermembrane space. This is the inner mitochondrial membrane. This is the matrix. Okay. From the ubiquinone, electrons are passed into the cytochrome B and proton to the intermembrane space. Then another molecule of ubiquinone also receive electron from FADH2. You know, FADH2 it is formed in the Krebs cycle when succinate to fumarate. When fumarate is formed from succinate, you know, FADH2 is formed. So, Krebs cycle take place in the matrix. So, from the matrix FADH2 is the oxidized FADH2 FAD. There the electrons are passed into the iron sulfur protein, electron and proton, and then the ubiquinone. From when it reaches to the ubiquinone, the proton is transferred into the intermembrane space, electron to the cytochrome B. Okay. Now, the cytochrome B carries the electron, it is passing to the iron sulfur protein, then to the cytochrome C1. So, the transfer of electron from FAD or FADs to FAD, then from the FAD it is transferred to the ubiquinone with the help of succinate Q reductase enzyme. But from the cytochrome B to the cytochrome C, it transfer it is with the help of cytochrome BC1 complex. As the name implies, cytochrome B and cytochrome C1, it is part of cytochrome BC1. So, cytochrome BC1 or complex 3 will transfer the electron to the cytochrome C. Okay. Finally, electrons are transferred from cytochrome C to the oxygen, final electron accepted with the help of cytochrome C oxidase, the complex 4. Here the cytochrome AA3 and copper, it is integral part of cytochrome C oxidase. It's very important. Remember, this cytochrome C and ubiquinone are mobile electron carriers which can freely move in the hydrophobic membrane. And don't think that the cytochrome C is here now. Cytochrome C is also present here. There is no space. That's why I have written this here. Cytochrome C and ubiquinone. That's a mobile electron carriers in the membrane. So, okay. The next process is oxidative phosphorylation. Oxidative phosphorylation is the process of formation of energy-rich compound called ATP with the help of energy released by the oxidation of reduced coenzyme. The enzyme for it is ATP synthase or complex 5, which is seen in the elementary particle. You know, the elementary particle, it is an elementary particle, usually you will say F1 particle. It has F0 and F1 particle. The head region form the ATP synthase, which is protruding towards the matrix. See here, a proton gradient is formed in the intermembrane space. How it is? You see, transfer of electron from one carrier to the other, when it passes, it's a downhill process. Energy will be released. With the help of energy, protons from matrix, here from matrix also, protons can be transferred. And also the proton present the NADH, that will be transferred or pushed into the intermembrane space. Thus, there is a proton gradient in the intermembrane space. It is a must for the formation of ATP. When these protons are passing to the matrix through the elementary particle or the F0 F1 complex, energy will be released. This released energy will be stored in the ATP. ADP become ATP with the help of energy and pyrophosphate. Now we can study about the ATP formation, how many ATPs are formed. Now we can calculate how many protons are pumped into the intermembrane space by the oxidation of the coenzyme. See, one NADH by their oxidation pumps 10 protons into the intermembrane space from the matrix, and oxidation of one FADH2 pumps 6 protons into the intermembrane space. Then I told you from the intermembrane space through the elementary particle, these protons should reach to the matrix. There, one ATP molecule is generated by the flow of 4 protons through the F0 F1 complex. When this 10 protons pumped by one NADP flows back to the matrix, 10 by 4, 2.5 ATP will be formed. Similarly, when the 6 protons pumped by one FADH2 flows back to the matrix, 6 by 4, 1.5 ATP will be formed. So this is the ATP production. It's a calculation of the ATP production. Okay. Next is, what is chemiosmotic theory? Is very important for question. It was awarded Peter Mitchell, and he was awarded Nobel Prize or also in 1978. See, movement of these protons through this F0 F1 complex is chemiosmosis, and the ATP production by this chemiosmosis is known as chemiosmotic theory. Okay. another aspect related to the electron transport chain is coupling. Electron transport is normally tightly coupled to ATP synthesis. Electrons do not flow through the ETC to oxygen unless ATP is simultaneously phosphorylated to ATP. There are certain uncouplers. Uncoupler 1, 2,4-dinitrophenol or DNP. Some chemicals like 2,4-dinitrophenol or DNP is uncoupling agent. It allows electron transport to proceed, hence oxygen is consumed, but ATP synthesis does not take place. This acts as an uncoupler by carrying protons or hydrogen ions across the inner mitochondrial membrane and hence dissipate the proton gradient. Hence, DNP is acting as an uncoupler. The second uncoupler, the thermogenin. Uncoupling also occurs naturally in some tissues. For example, the mitochondria of brown adipose tissue are uncoupled by a protein called thermogenin. The resulting heat is known as non-shivering thermogenesis. The heat produced by this adipose tissue serves to protect sensitive tissues in newborn animals and to maintain body temperature during hibernation. Another important aspect portion is electron transport inhibitors. See, there are certain inhibitors in the electron transport and oxidative phosphorylation. See, protinone and amytal, this will inhibit NADH dehydrogenase enzyme, complex 1. Then antimycin will inhibit cytochrome BC1 complex, and cyanide, azide and carbon monoxide inhibit the cytochrome oxidase. And these three affect the electron transport chain will be disturbed because these complexes are related to the electron transport. But oligomycin will inhibit the F0 component of elementary particle. Elementary particle consists of F0 and F1, head part ATP synthase, and then the F0 component is inhibited. So, oligomycin inhibits ATP production. So, remember, all these are related to or inhibiting the ETC, but the ATP production will be inhibited. So, now we can study or we can answer certain questions. Question number 1, the immediate energy source that drives ATP synthesis by ATP synthase during oxidative phosphorylation is the A. Oxidation of glucose and the organic compounds and other organic compounds. B. Flow of electrons down the electron transport chain. C. Affinity of oxygen for electrons. D. Hydrogen concentration across the membrane holding ATP synthase. Here the answer is very clear that the immediate energy source for the ATP synthesis is hydrogen concentration across the membrane. Flow of electron down the electron transport chain also may be the correct answer, but the immediate energy source is hydrogen ion concentration across the membrane. So, the answer is D. Second question, which of the following is a mobile electron carrier in the mitochondrial electron transport system? A. NADH dehydrogenase, B. FADH dehydrogenase, C. ubiquinone, D. succinate dehydrogenase. See, ubiquinone and cytochrome C are two mobile electron carriers in the mitochondrial electron transport system, you know. So, here the answer is C. Third question, cyanide, oligomycin and 2,4-dinitrophenol DNP are respiratory inhibitors in ETC and oxidative phosphorylation. Which of the following statements correctly describes the mode of action of three inhibitors? A. Cyanide and DNP inhibit the ETC and oligomycin inhibits the synthesis of ATP. B. Cyanide inhibits the ETC whereas oligomycin and DNP inhibit the synthesis of ATP. C. Cyanide, oligomycin and DNP compete with the oxygen for cytochrome oxidase, that is complex 4. D. oligomycin and cyanide inhibits synthesis of ATP, DNP inhibits ETC. We saw that cyanide inhibits the enzyme cytochrome C oxidase, the complex 4, and thus inhibiting the ETC, whereas DNP is an uncoupler and inhibiting the ATP production. Similarly, oligomycin inhibits the F0 component of the elementary particle and thus inhibits the ATP production. So, the answer is B. Last question, fourth. When electrons flow along the electron transport chain of mitochondria, which of the following changes occurs? A. pH of the matrix increases. B. ATP synthase pumps protons by active transport. C. The electrons gain free energy. D. The cytochromes phosphorylate ADP to form ATP. Here the answer is very simple. You know, when electrons flow along the electron transport chain, protons from matrix is transferred to the intermembrane space. So, decrease in proton concentration will increase the pH. So, the answer is A. That is all about electron transport chain and oxidative phosphorylation. I think this will help you a lot. Thank you.